Have you ever stopped to think about how a single cell, just one, can build an entire person? It's kind of mind-blowing, right? Every cell in your body, from a brain cell to a skin cell, has the exact same DNA, the same genetic blueprint. So how do they end up so different? Well, today we're diving deep into the cell's playbook to uncover the molecular tricks that a special class of proteins uses to control a cell's identity and ultimately its fate. So here's our game plan. We'll start with the big picture, meeting the cell's master architects. Then, we'll zoom way in on a tiny but powerful molecular switch involving a protein called SOX2. We'll see another one of these architects, SOX9, in action, and watch what happens when these two rivals go head-to-head. And finally, we'll uncover a brilliant strategy they use, something we're calling the cofactor heist.
All right, let's jump right in. Section 1, the cell's master architects. This is all about the central puzzle of biology, how one genome can create so many different destinies. So this is the big question. If the DNA is the master cookbook, how does one cell decide to bake a cake while another decides to grill a steak? The secret isn't in the recipes themselves, but in which pages of the cookbook are open and which are slammed shut and locked away. It's all about which genes are accessible. And this is where our heroes come in, the pioneer factors. Think of DNA in a cell as a library, where most of the books are locked in glass cases. Most proteins can only read the books left out on the table, but pioneer factors, they are the master locksmiths. They have the unique ability to go right up to those locked cases, pick the lock, and open the book for everyone else to read. They get in first. So how do these locksmiths actually work? Let's zoom in on one of the most famous pioneer factors, SOX2. It's a huge deal in stem cells. But what we're about to see is that even these master regulators have their own master switches that fine-tune their power. You see, a protein like SOX2 is the star of the show, but it can't do it alone. To really get things done, to turn genes on, it needs to recruit a posse, a whole crew of helper proteins called coactivators. The real question is, how does it choose its crew and get them to stick around? Well, researchers went looking for SOX2's partners, and they found that it really likes to hang out with a family of proteins called SRCs. And when they looked inside embryonic stem cells, they saw something specific. At all the spots on the DNA where SOX2 was working, they also found two members of this family, SRC2 and SRC3. But the third member, SRC1, it was nowhere to be found. This tells us it's not a random partnership. SOX2 is specifically teaming up with SRC2 and 3 to keep those stem cells ticking. Okay, so that's the who, but what about the how? How does SOX2 strengthen its group on these specific partners to really ramp up its activity? What's the signal? The secret, it turns out, is a tiny chemical tag called an acetyl group. It's something that gets added to the SOX2 protein after it's already been built. And you can think of it like a molecular dimmer switch. Adding this tag doesn't just turn the light on, it cranks the brightness all the way up. And here's the proof. When scientists added that acetyl tag to SOX2 in a test tube, suddenly its handshake with SRC2 and 3 became a super strong bear hug. The interaction was dramatically enhanced. And they pinpointed the exact spots on the SOX2 protein, these 3 lysine residues, where this happens. If they mutated those spots so the tag couldn't be added, poof, the enhanced binding was gone. And even more importantly, SOX2 got way worse at its main job, turning on genes. So this tiny chemical modification acts like molecular superglue, locking SOX2 and its crew together to get the job done. Okay, so that was a deep dive on one pioneer. Now let's pull back a little and meet another major player, SOX9, and see this pioneering ability, this lockpicking, in real time. This is so cool, you guys. Just look at this heat map. The blue signal shows where SOX9 has landed on the DNA. See how that happens at week 1? Now look at the orange signal. That shows when the DNA in that exact same spot actually opens up and becomes accessible. And that doesn't happen until week 2. There's a clear time lag. This is amazing evidence. It shows that SOX9 isn't waiting for an open door. It's the one that shows up first and kicks the door down. And that right there is the essence of being a pioneer factor. It gets in first, it does the hard work of opening up that tightly packed DNA, and it prepares the ground for all the other proteins that need to follow. It's the trailblazer. So now we have our two main characters, SOX2 and SOX9. They're both powerful pioneer factors, but what happens when they end up in the same cell? Well, it turns out they're often bitter rivals, fighting to push the cell in completely opposite directions.
A classic example of this rivalry plays out in lung cancer. Cells that are high in SOX2 tend to stick together, grow in neat sheets, and just divide like crazy, but they don't move around much. On the other hand, cells high in SOX9 totally change their shape, break free from the crowd, and become masters of invasion and movement. It's a fundamental trade-off. Grow in one place, or stop growing and start moving. And the choice comes down to which pioneer factor is in charge, SOX2 or SOX9.
This sets up a really fascinating puzzle. How does a pioneer like SOX9 not only turn on its own invasion program, but also shut down the old program run by its rival, SOX2? The answer is incredibly clever, and it's a strategy we can think of as a molecular heist. And this quote from one of the papers really nails it. To completely change who you are as a cell, it's not enough to just start doing new things. You also have to stop doing the old things. It's a two-part process. You have to build the new, but you also have to demolish the old. So how does SOX9 pull this off? It's all about its crew. Look at this experiment. On the left, you see normal, healthy SOX9. It binds to DNA and boom, the chromatin opens right up. But on the right, they used a mutated version of SOX9. They clipped off a specific part of the protein, the part it uses to recruit its cofactor crew. Now look what happens. The mutant can still land on the DNA, but it's powerless. It can't open anything. This tells us SOX9's pioneering ability is completely dependent on its power to bring in its helpers. And this gets us to the core of the heist. SOX9 doesn't win by directly attacking the old genes. No, it's much smarter. It wins by stealing the essential machinery, the cofactors that the old program needs to stay switched on. It starves the competition. And here's the footage of the heist in progress. These charts are tracking where the critical cofactors are in the cell. Before SOX9 is turned on, this machinery is busy working at the old gene sites. But as soon as SOX9 is activated, watch what happens. The signal at the old sites plummets, while a new, strong signal appears right where SOX9 has landed. It is literally hijacking the essential workers from the old factory and moving them to its new construction site. So you can break this whole heist down into 3 stages. First, in homeostasis, everything's normal, the old program is running smoothly. Then, stage 2, SOX9 arrives on the scene. It binds to new sites and starts luring away the cofactors. You can see the old program start to flicker and dim. Finally, in stage 3, the fate is switched. The hijacked machinery is now fully dedicated to the new program, and the old program, starved of resources, goes completely dark. And this model just puts it all together beautifully. You can see the old gene regulators losing their essential cofactors, which are being stolen and brought up to the new sites controlled by SOX9. It's not brute force. It's a brilliant strategy of resource competition that allows for a clean, efficient switch from one cell identity to another. Okay, so why does all this molecular detail matter? Because this process is happening in our bodies all the time. It's how we developed in the first place. It's how our tissues heal. And when it goes haywire, it's at the very heart of diseases like cancer, where a cell hijacks its own identity to become dangerous. If we can understand the rules of this cofactor heist, maybe we can find a way to stop it. And that leaves us with this final, really exciting question. We're seeing that cell fate is a battle for limited resources. If we can truly understand all the rules of that battle, could we one day step in and control it ourselves? Could we learn to rewrite a cell's fate to repair damaged organs or turn a cancer cell back into a normal one? We're just starting to decode the blueprint, but the possibilities are absolutely incredible.